We now have our next session, Antibodies against PURA A and PURB, making the invisible visible. Sabrina Bacher is a PhD student in the Niesing lab at the Helmholtz Center in Munich since 2020. She focuses on the cellular and molecular function of PURA and its implications in the neurodevelopmental disorder PURA syndrome. Already during her master thesis in the Niesing lab, she worked with induced pluripotent stem cells to study disease mechanisms of PURA syndrome. This work is now continued during her PhD. Before her PhD, Sabrina earned her bachelor and master's degree in biotechnology from the MCI in Innsbruck, Austria. She is highly interested in studying the cellular and molecular basis leading to the PURA syndrome in order to identify targets for future treatment strategies. Sabrina Burchick is a PhD student in the Institute of Pharmaceutical Biotechnology at the University Ulm in Germany. She studied biochemistry and pharmaceutical biotechnology at the University Ulm and the University of Applied Sciences in Biberach, Germany. Her current research focuses on cellular functions of the neuronal transport factors PURB and PURG and their interplay with PURA. She is particularly interested in the DNA and RNA interactions of PURB. Carolyn Kettler started her PhD in October 2021 in the Niesing lab at the Helmholtz Center, Munich. She has a bachelor's and master's degree in biochemistry from the Leibniz University and the medical school in Hanover. Her research focuses on identifying molecular pathways in the cell in which PURA plays a key role and which are impaired in PURA syndrome. Welcome to you all, and thank you for being with us.
Thank you, Damon, for the kind introduction. Just let me share my screen. So I hope that you can now see my screen. Nice, how it should be. So yeah, we are very glad to be here again this year. And Caroline, Sabrina, and we thought it might be nice to talk a little bit about antibodies and how we can use them to better understand PURA and PURB. So what will we talk about today? So first of all, I will give you an introduction to antibodies. Where do they come from? How do antibodies look like? And how can antibodies be used for our research? Later on, Sabrina will talk about how antibodies help to better understand the PUR proteins. So these are PURA, PURB, and PURG. And in the end, Caroline will talk about how all of this looks like in PURA syndrome patient cells, and also how we can use antibodies to better understand PURA's interaction partners. But let's start with the introduction first. And I guess after 2 years of Corona pandemic, everyone has heard about antibodies. But what also became more clear is that in the last 2 years, we became more aware about the fact that we are really constantly exposed to harmful germs, such as bacteria or viruses, such as the coronavirus, but also parasites and fungi. We are constantly surrounded and attacked by these kind of organisms. So now there's the question, why do we not get sick all the time? And that is because we have our own shield inside of our body that helps us to fight off these invaders. And that is our immune system. So our immune system is a very complex defense mechanism that actually starts with a physical barrier that is your skin and also the mucous membrane that covers your inner organs. And it ends with all of these very complex and many cells that help to fight off in the inside of your body if some germ gets somehow into your body. So I will not explain in detail what each and every one of the cells is doing. I just want to point out the one cell that is important for us today, and that is the B cell. So the B cell is one of the key players of our immune system. Why? Because it can produce antibody, and it starts with doing this with only one antibody that is stuck in its surface like that. So what is happening if you get infected? And let's say with a virus or coronavirus, for example. So the virus gets inside your body and it looks like this. So you see these little spikes here coming out of the surface. And these spikes are actually small proteins. And because they are stuck in the surface, we call them viral surface protein. So what is happening? This virus is floating in your bloodstream and tries to find the next cell to invade. And at one point, it might encounter a B cell with this bound antibody. So what is happening? The B cell is patrolling your bloodstream and actually looking for these kind of viruses. So it immediately gets suspicious about this intruder and wants to find out more about it. And how does it do it? It tries to bind to the virus. So the antibody here that's stuck in the B cell tries to bind to the viral surface protein. But this binding here can only happen if they fit perfectly. So what is happening if they fit perfectly? Then the B cell actually knows that this is an intruder that may cause us harm, and then it gets to work, basically. And that includes producing lots and lots of antibodies. And this is the same antibody that is stuck here in its surface, and it produces it over and over again, like a little antibody factory, basically, and it releases these antibodies into your bloodstream. And now they are freely floating around. So this is actually very important for a proper immune defense. Why? Because the antibody's job is to capture the viruses. And you see that multiple antibodies can bind to one virus. And that is actually very important because that prevents further movement and also further spreading of the virus in your body. And therefore, it also prevents spreading of the infection. So let's have a closer look at this antibody. And you see immediately that it has a very distinct shape. It actually looks a little bit like a Y. So we have a stem part of the antibody and we have two identical arms here. And I want to point out this upper part of these arms because this is where the binding to the viral surface protein is happening. And that is also why we call this binding site. And one antibody has two of these binding sites and they are identical. Now, the very special thing about these antibodies is that this binding site here is very specific towards its target protein. So what does specific mean? Let's head back to our example. So we have the virus here, so the coronavirus, for example, and this viral surface protein, and we zoom in. And you see that it actually looks a little bit like a key. And I have done this on purpose, of course. And I want to also point out the shape here in the front of the key. And now let's have a look at the binding site. Also, I want to point out the shape here. And I want you to imagine this binding site like a lock. And now you might also already imagine what is happening next. So the viral surface protein binds to the antibody at the binding site, and you see that they fit perfectly. So the two shapes, they really intertwine perfectly, and that is what specific binding means. So they fit perfectly due to their shape. And that means that only one antibody recognizes one protein. So a little bit like a key, and you really need the correct key to open the lock, otherwise it will not open. And that is actually a scientific term for us. So this principle that only one antibody recognizes one particular protein is actually called lock and key model or lock and key mechanism. But let me give you another example of a specific binding, which I also found quite good, and that is Cinderella and her glass slipper. So the slipper only fits Cinderella, not any of the other women that tried it on in order to get the prince. So this is the same basic principle. Now, the fact that one antibody recognizes one particular protein, that is what makes antibodies so interesting for us when we do research. So at some point, there were very smart people, smart scientists who understood what is happening here. And they thought that this is something that we can actually use ourselves. So they basically stole the idea of the immune system and thought that this interaction here can be true for any other kind of protein. So the specific binding. It's not a matter if it is a viral protein or a bacterial protein or any other kind of protein. So what scientists did, they started to design and produce antibodies with a specific binding site that fits to their protein of interest. For example, PURA. So there is the question, is there an antibody that only recognizes PURA? So you see here a specific binding site for PURA. And the answer is yes, there is. We work with such an antibody. And at this point, I really need to say again, or really make it clear that this antibody that binds to PURA is only used for research purposes. It cannot be used in any way to somehow treat PURA syndrome because PURA syndrome is not an infection. So we can only use it to better understand PURA. Now, how do we get an antibody against PURA? Because PURA is a human protein, we do not produce antibodies against it in our bodies. It's not a foreign invader. So the antibody the immune system will not react to PURA and build an antibody. So what we basically do is we borrow the immune system of a different organism. So what you need to have is when you want to produce a specific PURA antibody is you need PURA, that is the protein structure that you already saw in Dirk's video, and you need this other organism. And most of the time we use either mice, rats, or rabbits. So what we basically do is we inject the human PURA protein into the mouse's bloodstream. And what is happening, because this is a human protein and not a mouse protein, the immune system of the mouse will react to PURA in a similar way as it would towards a virus. So it treats human PURA as a foreign invader and wants to fight it off. So what is happening, the immune system of the mouse starts to work. And this includes also a B cell that binds to human PURA and produces antibodies that bind to PURA. And these antibodies we can now purify from the blood of the mouse and use it for our scientific work. Now, what can we do if we have this very specific antibody against PURA? There are several techniques which are based on antibody protein interaction, but I only want to talk about two of these techniques today because these are the techniques that we use in our lab mostly. And the first one is called immunoprecipitation. It's a very complex word, but what I wanted to know is that this is a technique with which we can pull PURA out of cells. Why do we do that? Because this allows us to find out what other things are bound by PURA. The second technique is called immunofluorescence. And this is a technique which makes the invisible PURA visible for us. And with that, we can answer the question where PURA is located in cells. So let's start with how immunoprecipitation works. How can we pull PURA out of cells? So we start with cells, of course, and let's zoom in again. So you see here a cell, this is the nucleus, and this is the cytoplasm, and you have this outer barrier here. And in the cell, there are lots of different proteins, and amongst them is PURA. So what do we need to do to somehow get PURA out of the cell? the first step is basically to destroy this outer membrane here. So we basically mash the cells up and put the remains into this liquid in this tube. And now all the proteins that have been inside of the cell are floating around in this liquid, including PURA. Now, how can we somehow pull PURA out of this liquid? That is where the antibody comes in. So we basically just add the antibody to the liquid here, and then the antibody does its job. So it catches PURA, binds to PURA. And what we can do now, we can somehow hook onto the antibody and pull it out of this liquid. And because the antibody binds to PURA, we also pull out PURA. And that is how we get, in the end, PURA alone. So this technique is most of the time not just used to pull PURA out of cells, but mostly it is used to find out what other things are interacting with PURA, namely proteins. So how does it work? Let's get back to that picture. And we are again in the liquid, and the PURA antibody here binds to PURA. And you see that there is another thing, another protein, which is somehow attached to PURA. So basically, PURA has found a friend, and they hold on to each other very tightly. So what happens if we come again with the hook and try to pull the antibody out of this liquid? And remember, PURA is still holding on to his friend. So what is happening? We do not only pull PURA out of this liquid, but also PURA's friend, so the protein, the binding partner of PURA. And with a very complex method, we can identify this binding partner, so PURA's friend. And if we know what PURA's friends are doing, we can also conclude what PURA might be doing. But Sabina will talk a little bit more about that later. So now you have heard how we can pull PURA out of the cells. But I also want to explain how we can make PURA visible when it's actually invisible for us. Let's have a closer look. So we start again with our cells, with all the different proteins and PURA. Only this time we are much more kind to our cells, and we will not destroy them. We only want to look at them. So we basically want to know, where is PURA in the cell? And now there is the first problem, because cells are very tiny and we have to look at them through a microscope. And these proteins, which are inside of the cell, they are even smaller. So we cannot see them just by looking at the cell. That is just not possible. So what we need is for PURA to give us a signal so that we know where it is. You can maybe imagine this like the friend that stands in a crowd and waves to you so that you can see him. Or you can just watch this very nice example of how this works. This is a scene from Harry Potter and the Goblet of Fire, where Harry is in the maze at the end, and he shoots these red sparks up in the air so that Dumbledore knows where exactly he is in the maze, although he cannot see Harry directly. And that is basically how this technique is working. So PURA is giving us a signal similar to Harry with the red sparks. So how exactly is this working? Let's zoom inside the cell where there are lots of other proteins and where there's also, again, PURA. So what is happening? We need to give PURA basically a wand so that it can give us these red sparks. And that is the antibody again. So we add the PURA antibody and it does what it should do. It will bind to PURA. Another clue about this technique is that we can add a very tiny glowing molecule to the stem of this antibody. And as soon as the antibody binds to PURA, this glowing molecule actually starts to glow. And that is something that we can observe in cells. So you see here, everywhere where PURA was before, or where PURA is, you see now this glowing signal, so the light signal, just like Harry with the red sparks. And the really cool thing is that we can not only observe this, we can also prepare or make pictures of that. And that is how it looks like. So we see here 2 cells, and you see lots and lots of tiny red dots. And each one of these tiny red dots here is actually a PURA protein that signals us where it is with this glowing molecule or with the red sparks like Harry did. So let me shortly summarize what I have told you so far. I told you that B cells are key players of our immune system because they can produce antibodies. Antibodies, their job is to capture harmful germs and prevent their further spreading. They have a very distinct Y shape with specific binding sites for only one protein. And this fact that only one protein is recognized by one antibody can be used in research. Antibodies for research are not produced in humans, but in animals. And there are several techniques that use antibodies as a tool. For example, you can use antibodies for pulling PURA out of cells so that we can find out more about PURA binding partners. And we can also use antibodies and a light signal to find out where PURA is located in cells. So I hope I could give you a good overview of how antibodies are produced, where they come from, and how we can use them. I would like to thank you very much for your attention. And now I would hand over to Sabrina.
Okay, thank you, Sandra. Now we'll try to share my screen and start the presentation. Okay, so hello and welcome everybody also from my side. I'm really happy to be here this year again and present you some of the work we are doing currently and tell you how we use these antibodies to better understand PURA syndrome. In the beginning, I want to refer to the presentations which we had last year, because it's then more easy to understand the stuff we are talking about today. So last year we had a talk which was called Stem Cells and Beyond. So there we talked about how we use induced pluripotent stem cells and also CRISPR-Cas9 to understand PURA syndrome better. And then we also had a second talk, and this was like a really general talk about genetics and what is Mr. PURA actually doing in the cell. So in a really simple way, we try to show you what we do to find out what PURA is involved in function-wise. So today I tell you where PURA is actually located in the cell by using these antibodies, which Sandra explained previously, and then also what is bound by PURA and what we also know about PURB. And I will start to tell you where is PURA located in the cells. Okay, here you can see like the human body. And as you might know, we don't only have like one cell type in the human body, but a lot of different cell types. So for example, we have brain cells, which look under the microscope a bit like this. And then we have also muscle cells, we have skin cells, bone cells, and blood cells, for example. But there are a lot of different subtypes and also different cell types, not only the ones I showed you here. So at first we ask ourselves what cell type we need now to better understand PURA and PURA syndrome. And as we know, it's a neurodevelopmental disorder. That's why we first wanted to focus only on neuronal cells to study this disease. And here you can see only some of the subtypes of neuronal cells. So as I said, there's not only one brain cell type, but a lot of difference, as here indicated by different colors and shapes. And these are also organized in a different way. So here in the outer layer, so this is like a cut through the brain. And here in the outer layer, we have a different kind of cell type compared to the inner layer. So now what we wanted to know is how PURA is localized in these cells and how it is also expressed there. So how much PURA do we have in the different kind of cells? And for that, we use the explained antibodies. So what you can see here is a microscopic picture. So here in blue, we have the nucleus. And so this is like the inner part, which was explained before by Dirk. And then here in red, visualized via antibody, we have PURA. So you can maybe appreciate here that PURA is mainly localized to the cytoplasmic regions, because here in the nucleus, you cannot really see a high signal. And as we zoom in, you see it even better. So just imagine here, this is one cell. And around the nucleus, we have a lot, like a high signal. And this is how we know that there should be a lot of PURA. And also we have this dot part. So looking like some kind of granules where there is a lot of PURA. And also PURA is not only localized around the nuclear parts, but also really far away from the cell body in these extensional parts. And this are indicated here as well. So we have PURA localizing around the nucleus, and also in the extensions where we see it as some accumulation in some spots. So PURA functions there and has maybe an important role at these different parts. And as I told you, there are many different kinds of neuronal cells in our brain. And that's why we also looked at different cell types, neuronal cell types. So here you have the stem cell. And from this stem cell, you can generate all different kinds of neuronal cells. That's why I refer to the previous talk, which we had last year, because then there I explained it a bit better. And here you can see this antibody staining, and in white, there is PURA. So here you can see that there is really less signal in the stem cells, meaning that there is really less PURA protein expressed in the cells. But then we have higher signal in these neuronal cells, meaning that maybe there the function of PURA might be more important than in the stem cell state. And like I showed you before, also in this different kind of neuronal cells, we see the same localization. So we have here this granule dots and also the localization around the nucleus, and not so much in the nucleus, but to some extent we see also a light signal here. So maybe there could be also a function in the nucleus, but mainly it localizes to the cytoplasm and also to these granules, which is really interesting. And then we wanted to know what is bound by PURA, to maybe get a better idea what the PURA function in the cells could be. And like Sandra already explained, for that we use this pull-down assay. So the input here in this small tube we have is just crashed cells. And then when you like magnificate this, what we have inside are a lot of different proteins floating around and also PURA. And then we use these PURA-specific antibodies to pull PURA down, and also, like Sandra explains, all the friends which are hanging out with PURA. And then these proteins, like the binding partners, can also be pulled down by just pulling this PURA antibody out of the solution. So with this bit more complicated technique, we can now identify what proteins are actually bound to PURA. And by that, we can generate some kind of network. So here just names, you don't have to remember this because these are just the binding partners we identified, and these are the names of the different proteins. And then you can generate a network, meaning now you know which proteins PURA interact with. And some of these proteins have already a known function, meaning when PURA is like a binding partner to these proteins, maybe it's involved in the same process. So this gives us really now an idea what process or what different kinds of processes PURA is involved in, and together with these other proteins which are bound to PURA. So now we And by identifying PURA's interaction partners, we could get a better understanding about the function, because here we have this huge network which have like one function, like all different proteins work together to like sort it out and do their part. And PURA is one part of this big process, but there are also different kinds of process PURA could be involved in, because this is what we saw looking at the different interaction partners. And the last point I want to talk about today is PURB. And also, like the question popped up before, I want to quickly explain what PURB and PURG or PURB actually is. So PURA is part of the purine-rich element binding protein family, and this is just like the abbreviation, so PREB. And these other proteins are part of this family. And why are those proteins also part of this PUR family? Is because they share a high sequence similarity, so they are really, really similar. And they also have a similar size. So for example, PURA and PURB share a sequence similarity of 70%. So they're extremely identical. And PURG also have like around 50% sequence identity, meaning they could have a similar function. And we also know, because of this pull down assay, that PURB binds PURA, and also PURA binds PURG, the different types of PURG. But today I only want to focus more on PURB, because there we also have an antibody, and we know a bit more than to the other protein. So as I showed you before, this is a microscopic picture. Here you can see in blue again the nucleus part, where we just like want to identify the cells. So here we have one cell, and here with the PURA antibody we see how PURA is localized in these cells. And again, you have these granular parts and this inner part where the nucleus is, and also the DNA, and there is not so much PURA. And now we have a PURB specific antibody, which was found by Sandra actually, and she tested a lot of difference, and now we have the specific antibody which only binds PURB. And here in green now you can see where PURB is localizing, and it's a bit different like PURA, which you can see. So PURB is surprisingly localizing more to the nucleus, but also a bit around the nucleus. And here we overlaid like all the channels, and this is, you can see here, and also in the zoom in a bit better. So you can see that in the cytoplasm, so around the nucleus, we have a lot of PURA and also PURB, but in this nuclear part, there is mainly PURB. And this, like I tried to show here a bit better if you don't recognize anything here or not a cell. So here we have PURB in this nucleus part, but also a bit around the nuclear part. And as we know, PURA is more in the cytoplasmic regions and here in the dots. Okay, and now as we know where PURA is localized, we can also, and where PURA is bound to, we know a bit more about their function, because as PURA is mainly in the cytoplasm, the function or the process PURA is involved in should be also here in the cytoplasm. And PURB maybe has more of a nuclear role. And if you maybe remember the slide Matt showed before, he showed that PURA back then was more associated to a nuclear function. And now we saw that it's more having a function here in the cytoplasm. So maybe all the stuff which was published earlier is actually true for PURB, because there we see this nuclear localization, and which also hints to a function there. Okay, to sum my whole talk up, so we know that PURA localizes mainly to the cytoplasm in the different neuronal cell types. PURA binds proteins, which which has known functions and associated with granules and also transport of RNA. And PURB localizes rather to the nucleus. And we can also find out a lot of more with these antibodies, but I don't want to go in so much detail today. Okay, and here what you can see is the PURA network. So here are just some people who are involved in the PURA research. In gray here, these are the alumni. You maybe know Lena and Marcel. And here in green, the active people from the Niesing lab, and also Melina, which is from the SANAC lab. And as I showed you before, the network of PURA, like every protein in this network has their own function. So do we in the PURA lab and in the PURA research. And maybe with one person, the whole process would not work, maybe without Dirk. And this is exactly what's happening with PURA. So one process in a cell is not working actually fine because there is no PURA. We didn't identify which process actually, but this is how you can imagine it. Okay, and now I want to hand over to Caroline. Thank you again for your attention.
Okay, hello everybody. I hope you can see and hear me, and you also see my slides and the laser pointer. So also warm welcome from my side today. So my name is Caroline and I'm a PhD student also in Dirk's lab at the Helmholtz Center in Munich. And I started my PhD at the end of last year. So I'm really new to this community and I'm very happy to be here with you today. And you heard a lot about PURA as antibody before. So what are antibodies, why do we use them, and how we can use them for our research. And what I want to focus on is, first of all, we will have a look at patient cells and the localization of PURA in these cells. And the second part of this presentation focuses on the co-localization of PURA and some interaction partners. So let's get started. One very important part of our research are patient cells, because patient cells have the same genetic background than patient cells in the human body, right? So maybe you know that we've already collected some skin cells from patients and we can cultivate them or culture them in our lab. So this is super nice. We have them now in our petri dishes, and you have to take really care on these cells. So you have to feed them. They need some food, and in this case, like media. And then you can grow them. They need enough space to grow. They need the right temperature. So it's a bit like having a pet or something to take care of cells like this. And you can do that. And when you do it right, and they are living cells, then you can do some nice experiments with that. And to do that, you also have to think about a nice control, because you always have to compare something to the patient cells. And that is, in this case, healthy skin cells. So we have here our patient cells. And you see that the PURA is here mutated in one of the copies. So we have a plus and a minus. Minus indicated the mutation in the PURA gene. And we have here, on the other hand, healthy cells where both of the copies of PURA are intact. And that's what you can use now, that we compare both of these cells. And you can do a lot of experiments with that. But what we focus on is, first of all, to check the localization of PURA in these patient cells. And you've already seen some nice images, and we will look at these skin cells now. So we have the healthy cells, which are here in the first row. And on the bottom, we see the PURA patient cells. And this is really like the same part of the slide when you're comparing these three images. They're just different, we call it channels. So we have like a blue channel where we see the nuclear staining. We have a red channel where we see the PURA staining. And in the end, we merge both of these channels to see the red and the blue signal in once. And what you can see here is nearly the same as what Sabrina explained already to you, because we see here in the nucleus where the blue staining is, we don't really see a signal for PURA, or really weak one. And but when you have a look at the cytoplasm, then you see a high signal here for PURA, and especially here in the tips of the extensions, there's a really high signal. And when you compare this now to PURA patient skin cells, you don't really see a difference. So for sure, every cell is looking a bit different, but when you compare the localization, then you also see that there is a weak signal for the nucleus and a high signal for the cytoplasm, and even higher here at the tips, at the end of the cells. So this is illustrated here as well. And for the initial experiments we did with the skin cells, we don't really see a difference. But that does not mean that there is completely no difference in localization of PURA in patient cells. But we really want to have a look at other cell types, for example, the neuronal cells. We come to that later. First of all, I want to give you an example what we are searching for, what we are looking for, by introducing you to another disease. And this disease is the neurodegenerative disease, and it's called amyotrophic lateral sclerosis. Maybe you heard about this disease before, but if not so bad. We have the ALS, and it can be caused by mutated PUS gene. So when you have PURA on the one hand, you have a mutation in the PURA protein or in the PURA gene, leading to a loss of the protein or a defect protein. And on the other hand, you have a mutation in the PUS gene leading to the ALS. So it's a really different disease, but what you can see, or what I want to show you here, is that we really see a difference when we look at healthy and patient cells. So in healthy cells, we see here that PUS is really localized to the nucleus. So we don't see any staining here. So the green staining would be PUS, and we don't see any staining here for the cytoplasm. But when you compare that to patient cells, we definitely see that not only in this nucleus is this green staining, it's also here surrounded in the cytoplasm. So we definitely see mislocalization of mutated PUS from the nucleus to the cytoplasm. So here we have the intact PUS protein localized to the nucleus. Then we have the mutated one indicated by the star here, and this is mislocalized in the cytoplasm. And we're not looking exactly for this, but this is something like a difference. This is something like a phenotype we can find out in our research. So we are looking for something like that to see some differences between patient cells and healthy cells. You've seen this image before when Sabrina introduced you to the different cells of the human body. And indeed, different cells have different jobs in human bodies. We have brain cells, muscle cells, skin cells, bone cells, blood cells, and many more. And each cell has a different job in the whole body. And you can also see that they have different shapes, right? And often this is related to their function. And we have looked at the skin cells. When we remember, we have the healthy cells compared to the patient skin cells. And we didn't see any difference for the initial experiments. But what would be super nice is to look at neuronal cells, because we want to focus on these cells, as Sabrina said, because we have a neurodevelopmental disorder, and we think that the biggest difference can be here in these neuronal cells. But we don't have them, we cannot extract them or something, and we don't want that. But there is a trick or something that we can somehow produce these cells out of these cells. And maybe this figure is familiar to you because it was shown last year in the PURA conference when Lena and Sabrina were talking about induced pluripotent stem cells. And you've seen this one before, that skin cells from the patients can be collected, and we can cultivate them then in the lab. And what would be the next step when we want to go to these neuronal cells, as I said, we have to go back a stage. So we have to go back to these so-called induced pluripotent stem cells, because they have the ability to go into any of these directions, so to be any cell of the human body. And this is not super easy, because the skin cells are super specialized cells, and they are happy with being a skin cell, and they don't really want to go into this direction. So you need a lot of power, and this process is called reprogramming. So you go back one stage, and you have to put some chemicals on there to reprogram the skin cell into an induced pluripotent stem cell. But this is possible. And when you have these cells, then you can go in any of these directions. And what you always have to consider, as I told you before, you always have to compare your patient cells to healthy cells. So you can do that with your patient skin cells, but you can also use healthy iPS cells to differentiate them into brain cells, for example. And here we have that. Here we have the healthy cell. We have our patient cell, and which is also super interesting to compare that to a complete, we call it knockout. So when both of the copies of PURA are affected, and not only one, which is the case for the patient cells. Because then when we see some difference, then we expect that this difference is even bigger than the difference from the patient to the healthy cell. So yeah, that's what we can do and what we're doing actually at the moment, because Sabrina is taking care of this part here to have a look at the differentiation from the iPS cells to neuronal cells. And when we have optimized that, we can have a look at these different cells, and maybe this give us some hint what is different. Okay, so that ends my first part, and we will now focus on the co-localization of PURA and its interaction partners. You've seen this image like in Sandra's and Sabrina's talk, because they introduced the method to you already, which is called immunoprecipitation. So just quickly, we have here our PURA antibody. We have PURA bound to the antibody, and some other proteins are also bound to PURA. And we can identify these proteins. Then we go to this step to have like a list of these proteins which were attached to PURA. These are the so-called potential interactors for us now. And what can we do as well when we know these proteins, because they have names and they have been analyzed before? We can now look at co-localization. So we can zoom in the cell again and have a look if they are in the cell at the same location. And to do that, for sure, we need antibodies again. Surprise, surprise. So we have here our interactor and our PURA, which are in the cell, and we can't really see them. And now we can find these both proteins with antibodies. So the PURA antibody for sure recognizes the PURA protein, and there are not only PURA antibodies, because there are also antibodies against different other proteins, for example, for this interactor here. And then they are attached to a glowing signal. So we have a yellow signal here for the interactor, and we have a red signal for the PURA protein. And when you now have a look under the microscope, then you can see both the yellow and the red signal. And maybe you remember that from like painting or something. Then you mix some colors like a red signal and a yellow signal, and you mix these both colors, then you get an orange one. And that's what we are looking for. So we're searching for orange areas in the cell, and to check if they are at the same location. And that's what you can see here. So we have healthy neuronal cells here, and we have the different, we call it channels. So the different signals, the blue signal again for the nucleus, then we have the red signal again for PURA, and now the new signal is the yellow signal for the potential interactor. And here, just all of these channels are merged. And when you have a look at these images, then you can see the same localization as we've seen before for PURA. But what is now interesting is that you see these like hotspots also in the yellow channel, because when you look at here at the, I hope you can see it, that there is like a really hotspot of this signal, the red signal, and the same is here, can be found here in the yellow channel. And when you merge these channels, this ends up with an orange signal. And this is super interesting. Maybe you can better see it here. It's just the end of the cell. So that's just the extension. The cell would go like this here. But here you see like these both dots, and you see them here in yellow as well. And you see them here in orange again. So that means that the PURA protein and the potential interactor are located at the same areas in the cell. And what can that tell us about PURA? So maybe both the PURA protein and the interactor are involved in similar processes, because you've seen an image of the cell, and there are a lot of compartments, and it's really well structured. And it's also that processes are really special for some parts of the cell. So when you know that they are both at the same position in the cell, it's really likely that they are involved in similar processes. And as Sabrina told you, we know much more for parts of the interaction partners than we know for PURA. So we can learn something about PURA when we look at the processes where the potential interactors are involved. And that is illustrated here. So by this co-localization or by finding some interactors, we can definitely learn something about PURA. Okay, co-localization and what comes next. So first of all, we have to confirm PURA's interaction network. You can see that here again, and you see all the different proteins are connected by these lines, and these are the interactions. But there are many different interactions, so the interaction can depend on, for example, other proteins or other molecules. So it's not that just two come together and are fine, but it's just that maybe they need somebody else to have this interaction. Or, for example, it can be weak or strong, the interaction. So you don't really know if it's a weak interaction or a strong interaction. And what you also have to think about is if it's transient or stable. So maybe they just come together in one of the processes in the cell, and then they split up and everybody's going its own way. Or they are like doing everything together because they are really best buddies and they don't want to leave the other one. And the second thing we are looking at for these protein interactors is what happens when you lose your best buddy. And you can imagine your best buddy in this case is PURA. And here we see like the interactor in yellow, we see PURA in red. And for example, these are like friends playing soccer like every day because that's their thing, right? And now what can happen is when you have like the mutation in the PURA gene, it can happen that you lose your best friend because he's like, for example, moving to another city, or he just changed so much that he's like more into tennis now and doesn't want to play soccer anymore. And I think you can imagine that this makes this interactor super lonely and sad because he lost his best friend. And some things are not really going nicely or as they're supposed to go when your friend's gone. So for example, when they played like soccer and every day they went to like a soccer field, then they are not going there anymore. So maybe the location of this interactor is also changed because PURA is not there anymore. Or the job, which is in this case like playing soccer, is not done anymore because PURA is not there. Maybe he also finds somebody else who wants to play soccer with him, like a good other friend, for example, PURB or PURG, but you don't really know. And I think it really has a consequence when you lose your best buddy here. Okay, last I want to sum up what I talked about today. And first of all, we looked at the localization of PURA in patient and healthy cells, and that with definitely go on because we just have this initial experiment with the skin cells. So we really want to look at different specialized cell types and especially neuronal cells. And then I talked about interaction partners and what they can tell us about PURA. And we talked about consequences for these interaction or binding partners in the cell when PURA is mutated or you have a loss of PURA. And I want to conclude with the sentence with the title of our talk. So antibodies will help a lot in the future to make the invisible visible. So without them, we don't have a chance to find PURA because there's so many proteins in the cell. And we introduced you to 2 techniques, but there are even more applications where you really need antibodies to tell you something, because we can't see them, we can't touch them. And we just need these little tricks to find out something about PURA. And with that, I come to an end, and I want to thank you for your attention. I want to thank the entire Niesing lab. You heard Sabrina talking, Sandra talking, and that was me. And we have like a nice group, and we have some people at the Helmholtz Center in Munich. We have some people at the university in Ulm. This is somehow confusing sometimes, but we are like one big group, and the Niesing lab. Then I want to thank Sebastian and Jona at the Institute for Stem Cell Research. They helped us a lot with the stem cells, and the Jona, for example, did the reprogramming from skin cells to induce growth in stem cells. Then I want to thank Melina and Kati at the University in Frankfurt. They did a great job, or are still doing a great job with the bioinformatics stuff. And then I want to thank the PURA Syndrome Foundation for having us here and for their support with our work. And yeah, if you have any questions, just let us know. And if there are some unanswered questions or they come up later, then don't hesitate to reach out to our email addresses here. And yeah, with that, thank you for your attention.
Okay, thank you, Caroline, Sandra, and Sabrina for your presentation, and your work is amazing and very much appreciated. We did have one question come through. If there's any more questions, please type them below in your Q&A at the bottom of your screen. And again, if you think of it later, you can always post it to our website for the Saturday panel. So one question came in from a parent. Just to be clear, there is no connection between PURA syndrome and ALS. Is this correct?
Maybe I can try to answer this question. So for me, it was just an example to show you that different localization can have an impact and can lead to a disease. But in fact, ALS is not that. It's also a neurological disorder, and somehow some papers are also focused on PURA's role there. But it's not that it's like it's super related, or that for now we can't use like the ALS or the FAS protein to know something about PURA. I don't know if somebody else wants to add something.
Another question came in. Have you found different amounts of PURA protein associated with different mutations?
So we just have 2 different mutations for the patient cells so far in our lab, and we, for the stainings you've seen, there's no really like you can't really check the levels, so you need some other experiments to find out some different levels. And we just have 2 different mutations so far, and but it just, for all of the other things, it seems that there is no different amount of PURA for different mutations, because we think that the mutations just lead to a reduced level of PURA. So we think it's more like that PURA is so defect that it's just not expressed, and we have like a 50-50 thing. And so I would expect that we don't see differences in the amount of PURA when we look at different mutations, but we will see different levels when we compare mutations or PURA patient cells to healthy ones.
Yeah, maybe I can add something to that. So actually it doesn't really matter, because we know that all the different mutations result in a not functional protein. So we know that 50% from the one chromosome is working and 50% are not working because of the mutation. And so these lower levels result in a disease. So it doesn't really matter the different amounts of protein, because actually like half, like 50% is like working and 50% are not working. So we would expect like similar amounts of protein, but one part is just not working and not like doing its function, which it should be, and resulting in a phenotype. Yeah.
Okay, another question has come in. Have you looked at whether stem cells can change or affect the mutations in PURA?
I'm not sure if I'm getting this question correctly, but we don't use stem cells to like have a look at patient cells or something. We just use the stem cells to differentiate them into in different kind of cells. So it's not that they help us to cure anything or to rescue a phenotype or something. So these stem cells are just for us, these induced pluripotent stem cells we get from the patient cells. These are just to differentiate them into different cells, and we don't use them like a treatment or something to rescue a phenotype.
Yeah, maybe we can refer to the slide Matt showed before, where you have these different model systems, and we are working with a human model system using these stem cells. So yeah, like Caro said, it's not like in treatment direction or so, but more to study the disease.
Okay, I thought I saw another question come through. I'm checking with them right now. Technology is amazing. So how many different proteins are in a cell? Do you know?
There are a lot of proteins. I don't know a number, but there are a lot of proteins, because basically all of the cytoplasm is tightly packed. So it looks like they're floating around, but essentially all of the space in the cell is used, and this is just full of protein.
And just for example, with the PURA screen, we already get thousands of different interaction partners. So it's not only 10 or so, but even more.
And then we have to find out which ones are just there by chance or are really interacting.
Have you stained cells other than skin or brain cells?
In terms of patient cells or in general? I guess in general, we tested different kinds of cells, not just skin and brain cells, but I guess also cells derived from the liver and the lung. But for patient cells, we only have these fibroblasts so far.
Have you ever done a Western blot to prove there is a reduction in protein levels?
I think that refers to the skin cells we have so far from the patients, but the problem there is that we need a control with the exact same genetic background, and I think this is something we are struggling with at the moment, because we have some healthy fibroblasts, which are kind of the same cells that we have from the patients, but they are not genetically exactly the same, except for the PURA mutation. And so when you do a Western blot on that, you can see something like that, but it would be very, not state out that, because we really need the same genetic background and the same cells, except for the PURA mutation, to really say that we have this reduction for 50%, which we would expect.
Okay. I'm not seeing any more questions. Oh, one just came in. If your hypothesis is that the mutated protein are just not made and only the wild type is made at reduced level, why then expecting differences in localization?
I think so. What we try to say is that we do not expect a different PURA level. We just expect that 50% of the PURA is somehow deformed and is not doing its job. But there should still be the same amount. Only half of it, it does not look like it's supposed to look like, and therefore cannot work properly, but it's still there somehow. And because it's not looking like it is supposed to look, one could assume that it just got stuck anywhere else in the cell because it's not functioning anymore. But we do not basically expect that they have differences in levels. Or does anybody else want to add something here?
Okay. I think that concludes the questions for now. Like I said, if y'all think of any more questions, please add them to the website. Thank y'all very much for your time and for being here.
Thank you very much.
The next session will begin at 4 p.m. Eastern time. It's feeding therapy and coffee with Rachel Rose. And thank you all for being here.
Bye.
Bye.